Hello, I'm Dr. David Wirta, a clinician and researcher at the Eye Research Foundation in Newport Beach, California. My interests include ocular surface disease and dry eye therapeutics. Dry eye disease is a condition of tear deficiency in which the body is unable to produce the quantity or quality of tears needed to maintain ocular surface homeostasis. Tears are made up of thousands of biochemical components, including lipids, which are important to slow tear evaporation. Acoltremon is an agonist of TRPM8, which is a key regulator of basal tear production. By stimulating TRPM8, trigeminal nerve activity leads to stimulation of the lacrimal glands, goblet cells, and meibomian glands. Alcon conducted a phase 3b study to characterize the tear volume and tear lipid concentration stimulated by Acoltremon ophthalmic solution 0.003%. This was an open-label, single-dose, single-arm study of Acoltremon 0.003% in 41 adults with dry eye disease. At baseline, optical coherence tomography, OCT, imaging of the central tear meniscus was performed in the left eye, and basal tears were collected from the right eye. 3 minutes after administration of 1 drop of Acoltremon 0.003% in each eye, tear meniscus imaging and tear collection were repeated. Key endpoints included change from baseline in tear meniscus height and tear meniscus area measured by OCT, and change from baseline in total tear lipid concentration. 3 minutes after instillation, Acoltremon 0.003% led to a statistically significant increase of 147% in tear meniscus height from baseline. Tear meniscus area also increased significantly, from over 51,000 square micrometers to over 200,000 square micrometers, an increase of 324%. 34 of 41 subjects provided a sufficient tear sample to analyze concentration of tear lipid. The geometric mean total tear lipid concentration increased from 128 nanograms per microliter at baseline to 221 nanograms per microliter after dosing, a 72% increase. Regarding safety, the only reported adverse event was instillation site irritation in 39% of subjects. All subjects experiencing this event rated the sensation as mild. In conclusion, a single dose of Acoltremon ophthalmic solution 0.003% resulted in statistically significant increases in tear volume as indicated by both tear meniscus height and tear meniscus area measurements, as well as increase in total tear lipid concentration. Lipids are an important component of natural tears, contributing to tear film stability and helping to retard evaporation of tear fluids. These findings further characterize the physiologic tear response associated with selective TRPM8 activation and are consistent with previously observed increases in tear production in pivotal phase 3 studies.